In this video, we're going to talk about how to turn ammonia, how to use ammonia to make amino acids in bacteria. This process is called nitrogen assimilation. These processes happen in all different other types of organisms, in fungi, in plants, in some animals, not in people, I don't think. But we are just going to talk about them in bacteria, but if you hear about them later somewhere else, do not be freaked out by that. So nitrogen assimilation is going to take the ammonia that was made either through nitrogen fixation or through assimilatory nitrate reduction and use that ammonia in the building of amino acids. There are two different ways to do this, depending primarily on how much ammonia is present. So if there is a lot of ammonia, then the bacteria will use this pathway, which is alternatively called reductive amination. I'll write this down on the next slide. To make glutamate, both of these make glutamate. If there is a low amount of ammonia, then the bacterium will use this system, which is called the GS-GOGAT system. And the low concentration system requires ATP, so the cell will not use it unless it has to. Both of these processes will make the amino acid glutamate, and then transaminases will be used to make other amino acids. I think I told you guys in a previous video that every amino acid has its own transaminase. I have since learned that that is not exactly the case. So there are groups of related amino acids, and each group of amino acids will have its own transaminase, but every single amino acid will not. So that was not totally correct. Every group of amino acids will have its own transaminase. Okay. So to make an amino acid from a high concentration of ammonia, again, is called the reductive amination pathway. Okay, can you guys see that? Yes. And so this starts with a carbon skeleton right here called alpha ketoglutarate and starts with ammonia. So we are going to talk quite a bit about alpha ketoglutarate. You guys know that chemistry is not my strong suit. And so I can talk like relatively well about these high and low concentration ammonia assimilation pathways, but I cannot draw for you alpha ketoglutarate if I needed to. I couldn't. So I am just going to call it alpha kg and learn and teach you how it is involved in these processes, but I personally am not that concerned about the structure. So if looking at structures really helps you, absolutely look at it. If looking at structures does not help you and may be discouraging, do not look at it. This is what I am going to use and what you need to know. So the high concentration ammonia pathway, we are going to have our alpha ketoglutarate, which is our carbon skeleton basis for glutamate, and then we are going to add ammonia. We are also going to add NADPH because we are going to be making a new bond and we need some electrons for that. And so this process makes the amino acid glutamate. Scoot this over a little bit. This is catalyzed by the enzyme glutamate dehydrogenase. This enzyme requires NADPH but does not require ATP. Okay, so that is what you can see here. I feel like these kinds of diagrams can be tricky to interpret because it has like the reactants and the products kind of looped together and they're confusing to me. And so I'm just going to draw it out for you separately. But if you like this kind, then here it is. Here is the text that goes with what I just said. This process makes glutamate, and then a transaminase will be used, and these are specific for the groups of new amino acids produced. The transaminase will other amino acid be used here to make some other amino acid, and then we'll regenerate the alpha ketoglutarate, which will come back up here. Let me zoom out a little bit so you can see that. Okay, so this is the high concentration of ammonia, high concentration of ammonia, which is also called the reductive amination pathway. This is it right here. Okay, so let me scooch off this one. Oh, then I have some text slide here for you. If you specific for the amino acid group, you see I have this error all over. I'll just fix it as we go. Okay, so if you need this slide, you can pause here, but we are going to go on. So there will be some times when there is not enough ammonia for the glutamate dehydrogenase reductive amination to work, and then the bacteria will do the low concentration ammonia pathway, which again is called the GS-GOGAT system. And you'll find this specifically mentioned in several different organisms, bacteria, fungi, plants, some animals. Okay, so just like the last one, I'm kind of confused by these diagrams, so I'm just going to write it out for you. So this is a two-step system. So the GS-GOGAT is referring to the two steps, so step 1, step 2. So in the first step, we actually start with glutamate. Let's see. Which I am going to, somewhat confusingly, write as alpha ketoglutarate amine group, because this is going to help us here in just a second. So we are going to start with the glutamate, and we are going to add more ammonia, and we are going to make glutamine, which is alpha ketoglutarate with 2 amine groups. Ta-da! This is done by the enzyme glutamine synthetase. Synthetase. T right there. Synthetase. This is super important because synthetases require nucleotide triphosphates. And so this particular one uses ATP. So glutamate plus ammonia catalyzed by glutamine synthetase gives us glutamine, and this reaction requires ATP. Okay, so now we have our glutamine, which is our secretly alpha ketoglutarate with 2 amine groups on it. So in the second step, we actually combine the glutamine with an alpha ketoglutarate to make us 2 glutamates, which is alpha ketoglutarate with 1 amine group. So basically, we just take one of these amine groups and move it over here to this alpha ketoglutarate. This reaction is catalyzed by the enzyme glutamate synthase. Do not get these backwards. Do not. So glutamine synthetase, which uses ATP, glutamate synthase, which does not use ATP, but does use NADPH. And so 2 glutamates are made. One of them is going to come back up here to be used to restart this process. And then the second one is going to be used with transaminases to make other amino acids by moving this amine group, which would then regenerate the alpha ketoglutarate to be used either here or in the high nitrogen, high ammonia process. Okay, so this is what happens when ammonia is not as prevalent in the system. Is the low ammonia process. It is a 2 steps, and the first step requires ATP. So there you have it. Um, so this, yeah. So here is kind of this bullet point. So again, the first enzyme is called glutamine synthetase, and synthetases use nucleotide triphosphates. This one uses ATP. The second step is catalyzed by glutamate synthase and requires NADPH. The end product is 2 glutamates. Okay. And here is a super text heavy slide. If you would like to pause here, you can. Okay. So for the last slide, I have this one. In case you need it, we are just going to go through and fill these in to make sure that you guys have something. You know how I like the summary situation. So we are going to do that here. So we'll start up here with nitrogen fixation. So nitrogen fixation is where we take, can you guys see? Yes. Nitrogen gas and turn it into ammonia. So this is done by the nitrogenase, you might write smaller if you were writing this on your paper, by the nitrogenase enzyme complex. This does not require NADPH. Since I last talked to you, I looked this up, and the mysterious electrons actually come from metal ions, not from NADPH. So no NADPH is used, ATP is used, and super importantly, this is inhibited by oxygen. I'm sure you guys will remember my excitement about the leg hemoglobin. And so in order for nitrogen fixation to be done, there has to be a way to remove oxygen from the area right around the nitrogenase enzyme. Okay, so this is one way to get ammonia. The other way to get ammonia is assimilatory nitrate reduction, which, as you can tell, is a two-step process. And so the first step uses nitrate, which is NO3 minus, and makes nitrite, which is NO2 minus. The enzyme that does this is called nitrate reductase. It uses NADPH but does not use ATP. In the second step, the cell will take the nitrite, the NO2 minus that was made in the first step, and turn it into ammonia. The enzyme that does this is called nitrite reductase, and it does not use NADPH or ATP. So once the cell gets ammonia, the process of incorporating ammonia with carbon skeletons to produce amino acids is called nitrogen assimilation. We learned 2 different ways to do this. Both of these ways make glutamate, and then transaminases, whoopsie-daisy. Transaminases are used to transfer the amine group to other carbon skeletons to make other amino acids. So if there is a high concentration of ammonia, then the cell will do reductive amination, which uses ammonia plus alpha ketoglutarate and makes 1 glutamate. The enzyme that does this is called glutamate dehydrogenase. Does use NADPH, does not use ATP, and this is not really a deamination or a transamination. It's mostly just an addition, because we are just adding the amine group right on there. All right, if the cell has a low concentration of ammonia, then it will use the GS-GOGAT system, which is a 2-step process. So in the reactants are glutamate and ammonia, and the products are glutamine. This is done by the enzyme glutamine synthetase, right? Super important T right there. That T helps us to remember that although this reaction does not require NADPH, it does require ATP. The second step uses the glutamine that we made in the first step plus an alpha ketoglutarate, makes 2 molecules of glutamate using the enzyme glutamate synthase. Does require NADPH, does not require ATP, and is a transamination reaction. This one is neither because it's just an addition. So hopefully this summary sheet will help you guys to keep all these straight.